Well, hello everyone, and welcome to the 9th edition of the AMPS Congress. I'll begin with a short introduction in French, then continue to English. Bonjour à tous, bienvenue pour cette 9e édition du congrès annuel de l'association Médecine, Pharmacie Sciences et la première édition virtuelle de notre congrès. Comme vous le savez pour la plupart, la MPS, c'est l'association française qui réunit les étudiants en double cursus en France, c'est-à-dire qu'ils font une formation à la recherche en complément de leurs études de santé. Et le congrès est notre manifestation annuelle dans laquelle nous présentons des scientifiques, médecins ou pharmaciens chercheurs afin qu'ils vous présentent leurs travaux dans la plus grande diversité possible. Ci-contre, vous pouvez voir le programme prévisionnel de notre congrès, qui va s'étendre sur l'ensemble des week-ends du mois de juin. Et vous avez également la date de notre assemblée générale qui se tiendra cette année le 27 juin à 10h et qui est ouverte à tous les adhérents. L'adhésion à la MPS, je le rappelle, cette année est gratuite. Vous avez seulement à vous enregistrer sur notre site internet. Then, welcome all for first virtual edition of the AMPS Congress. AMPS is the French MD, PhD and PharmD PhD Students Association. And for this Congress, we will have MD, PhD and PharmD PhD researchers which will present their works during all the weekends of June. Here you can see on the screen the provisional program, and then I give the speak to Jeremy, which will present our first speaker. Thank you.
Okay, sorry, I had a problem. So I'm sorry, I will introduce, and now I have the honor to introduce Professor Gisella Vetere from the ESPCI in Paris. Is it working? So Professor Vetere did her PhD in Roma before pursuing a postdoc position at the Toronto Children's Hospital.
Yes, we can hear you.
She's now working in the C4 team at the ESPCI, named Cerebral Codes and Circuits Connectivity, currently working on the acquisition and consolidation of memories in mice and neuroplasticity. So I let the Professor Vetere talk to us, and then we'll get the question at the end. Don't hesitate to ask questions on live on YouTube, and then we'll take them and transmit them to Professor Vetere. Thank you very much.
In our laboratory at the ESPCI in Paris, in the C4 team, we are studying cerebral codes and circuits connectivity. And what we are interested in is to understand if there are cerebral codes that the brain uses to process external information. And secondly, we are interested in the circuits that the brain uses and how they connect to each other. The circuits that the brain uses to actually incorporate those informations. And in particular, we are interested in the study of memory. So why memories? Memories are one of probably the most important skill that we have. Without memories, we will be unable to identify and recognize ourselves. To understand that we are living, basically, because we will not remember what we just were 1 second before. And we also, thanks to memory, learn from our mistakes, and we are able to act based on our past experiences. So memory is probably one of the most fascinating skills that we have. And that's the reason why we want to study how the brain processes those information and how the brain makes from an experience a memory. So which kind of memory do we use in our laboratories? Well, memories that we remember. For example, the talk that I'm giving right now, it will not probably be remembered for the majority of you it's sad to say, but you will forget it in less than 1 week. And if I'm lucky, if I'm good in my speech, probably you will remember it for longer time, maybe for up to 1 year or so. So the memories that instead we study in our laboratories are memories that can be remembered for longer time, maybe for a lifetime. For example, if today you will have your first date with the love of your life, or the love of your life will tell you that he or she doesn't want to stay with you anymore and wants to break up with you, you will remember today. So those are the memories that we are interested in, the memories that we are studying in our laboratories, memories that are linked with an emotional valence, that have a strong emotional valence. And one of the examples of those memories, the memories that we study, is the Pavlovian conditioning, a type of learning that was discovered from Pavlov in the 1890. And where he found that when he was associating a drill bell with the food, a dog was responding to the ring of the bell, even without the presence of the food. So basically, generally the dog is salivating when the food is presented. That is the most important reward for a dog. But when he was presenting the ring of the bell right before the presentation of the food, the dog was starting to salivate, even just at the presence of the bell. So the ring of the bell now represents a stimulus that is associated with the reward, with an emotional valence, and the dog learned to create a conditioned response to this stimulus that was previously a neutral stimulus. So in our laboratories, the experiments that we do are very similar to this one. So we use Pavlovian conditioning, and in particular, we can use Pavlovian fear conditioning or Pavlovian reward conditioning, like in the case of the dog. And in our case with mice, what we learn to associate is, for example, a specific odor that is represented by a neutral stimulus, the conditioned stimulus, with a series of food shock. When the mouse learned that the odor is associated with the food shock, now the odor will present a negative valence, an emotional negative valence, and when the mouse is exposed to the odor again, it will spend less time into the compartment in which the odor is presented. So the previously neutral stimulus become now an aversive stimulus, and the mouse will present this conditioned response that will represent to the avoidance of the odor that is associated with the shock. And as you can see in this graph, the mice will spend less time in the compartment in which the odor is presented. So what we are interested as neuroscientists is to try to understand what happened at the neuronal level when the mice learn to associate these different stimuli. So we are interested in understanding if actually there is a code in the brain that the brain uses to incorporate this external information. So what is the role of those cells that are active when the mouse is experiencing a neutral stimulus or an aversive stimulus? Can we actually study those cells, try to understand what is the role of those cells? But to really answer this question, we first need to know how the brain works. So here is a very interesting image that we can now take from mice, from the brain of the mice, using techniques like the IDs code that allow us to clarify the rest of the brain and tag only some subpopulation of neurons in the brain. And here you can see that we can clearly see subpopulation of cells and where they project to other brain regions. In fact, the brain is an incredible complicated circuit made of hundreds of billions of cells that are called neurons. And the brain is made of thousands of different types of neurons that have different shapes that are made of different molecules and that connect to different brain areas. Every one of these cells is an incredible electrical device. It receives inputs from thousands of other cells through the axons that connects cells to each other. And when it receives these inputs, it's going to compute these inputs to their own electrical output, which then, if they pass a certain threshold, we go to thousands of downstream cells. So this process, which takes just milliseconds or so, happens thousands of times a minute for each one of the billions of neurons, as long as we live, as long as we think, as we feel, as we act. So how are we gonna figure it out which one of these single cell and then which one of the single population of cells and finally circuits works? Well, eventually, the final goal of neuroscientists is to try to understand how the entire brain works and how it orchestrates its activity to incorporate external information. And eventually, if we could in the future be able to get such a map, if we could look at the organization of molecules and neurons and networks that are active to incorporate specific information, maybe we could really understand how the brain conducts information from sensory regions, mix it with emotion and feelings, and generate our decisions and actions. We could potentially use this knowledge to find specific targets for new drugs to repair the computations that affect patients with brain disorders, and in my case, patients that are affected with memory disorders. So the best way to really understand how a specific cell or a specific circuit work in forming new memories, we should be able to use these cells, these circuits, manipulate them to destroy and create new memories. So the human being had always have the desire, I would say probably the nightmare, that someone can manipulate its brain to induce unreal memories or to delete existing ones. So even if these that you see right now are sci-fi examples, the reality, I would say, is not so far from these movies. Well, really, of course, we are talking about mice. We are not talking about humans. Nothing at the moment is possible on humans, but in mice, we are today able to actually use different techniques to target specific subpopulation of neurons, tag them, and then inactivate and reactivate them to study their function in memory processes. And this is exactly what a series of researchers found in different papers, showing that the cells that are active when the specific Pavlovian conditioning is learning are very important for remembering the memory trace the day after. So, for example, in this case, when a mouse learned to associate a specific, like a series of food shock with a specific context, a subpopulation of neurons is activated. And using genetically engineerized mice, we can tag those cells, the cells that are active when the mouse is learning this specific task, and then reactivate them or actually inactivate them, delete them in the same context. So the mouse generally, when it's put back in the same context the day after, he generally has a freezing behavior. The freezing behavior is a very specific behavior that the mice have when they are afraid of something. And in this case, when the mouse learned that in that context it got a series of food shock, he will be afraid to get again those series of food shock. So what he does is to have this behavior of freezing that is he's behaving like being completely immobilized, And we can study this as an index of his memory trace. So when we inactivate those specific cells that were active to acquire the memory trace, we found that the mice are not freezing anymore. So it looks like the mice are completely deleting the memory trace. So different techniques has been used to prove that those cells are very important to recall the memory trace. And in particular, one of them is for sure optogenetics. Optogenetics was a game changer in neuroscience. And it starts very simply by some photoreceptors, some sensory photoreceptors that are in unicellular green algae that controls phototaxics. So basically, in response to light, these photoreceptors, they respond to light, and unicellular alga can move due to the presence of light. So if we express these specific cells, these photoreceptors in other these specific photoreceptors in specific cells in other organisms, we can enable light to control electrical excitability, intracellular acidity, calcium influx, and other cellular processes. So in particular, in specific, what we can do is to take these cells, these genes, the genes that are expressing the opsin, the photoreceptor that is activated upon the presentation of the light, and then incorporate these genes in a viral DNA, and then inject this viral DNA into the brain of the mice. And in particular, we can actually inject this brain in specific subpopulation of cells, and then just implanting an optic fiber right above the selected region, we can actually induce the activation or the inactivation time dependent of these specific cells. So we can actually control genetically defined neuronal population in real time while the mouse is behaving, is performing cognitive tasks, as you can see here in the example on the bottom right. So just presenting the light, we can see the change, the real-time change in the behavior of the mice, and study how a specific subpopulation of neurons impact in the behavior of the mouse in real time. So using this technique, a group from the Nobel Prize Tonegawa laboratory was able to identify and create artificial memories for the first time in mice. And in particular, the two main authors, the two main researchers involved in this study were Steve Ramirez and Xu Liu. And they found that it was possible using optogenetics to create artificial memories in mice. So what they did was very simple. When a mouse explores a box, so a specific context, and he receives a series of food shock, a specific subpopulation of cells will be activated in his brain. And then when he's placed back in the same box, the mouse will freeze because he's remembering of the negative memory that he experienced before in the same box. So using optogenetics and genetically engineerized mice, we can actually tag the cells that were active when the mouse is experiencing this aversive memory. So we can tag those cells and then reactivate them or inactivate them when the mouse is in a different context or in the same context. In their case, what they did was to activate those cells in a completely different context, when a context that was actually not at all associated with a negative emotion. And what they found is that the mice was freezing in this new context. And this means that they were able to create or at least bring the fear experience in this new context. in a way creating an artificial memory. So when I was in Toronto, I was always very excited about the idea of creating these artificial memories, because we can learn a lot by studying this memory and the cells that are activated when we form artificial memories. But in particular, I was very intrigued by the possibility to create a completely artificial memory, in a sense that all the experiments that were presented before always try to tag the cells that were active when a real memory was created, and then manipulate them, change them in a way that you can create a slightly different type of memories, but still using those those memories that were really created before. So in my case, what I really wanted to do is to actually prove that we can that we can create memories just knowing specific codes in the brain. What I mean with that is that if we really believe that there are codes in the brain that the brain uses to incorporate external information, we should be able to actually stimulate specific subpopulation of neurons and activate and create the formation of memories of events that were never experienced before. So in other words, we should be able to, if we really believe that there is a code in the brain, we should be able to implant false perception, emotional memories, just by substituting the external experience with the stimulation of those codes in the brain. And that's what I wanted to prove in my experiments. So to do this, we actually need to know a specific code in our brain. And to just give you an example, if we go to Google Maps and search for a specific region, like places in the world, we don't know a priori which cells will be activated in the hippocampus. So the hippocampus is a very well-known region in our brain that is necessary for incorporate spatial information. So we don't know, though, when we are experiencing a new context, which cells will be activated to incorporate this spatial information. Differently from the hippocampal system, the olfactory system is very useful for our purpose, because we know exactly for each odor that the mouse is experiencing at the moment, which neuronal subpopulation of cells will be activated in its olfactory bulb. In fact, there are maps of activation of the olfactory bulbs available online, specific for each single odor that the mouse or a rat is experiencing. So this is a very unique system that allows us to actually know a priori which will be the map activated in the specific brain region that is the olfactory bulb when we are experiencing an external stimulus like an odor. And this is possible in the olfactory system because of the unique type of the system, of how is this olfactory system formed. So just to give you an idea, I schematized here how the olfactory system works. So basically, every time that the mouse is experiencing a different odor, the other particles will be activating a specific olfactory sensory receptors that are expressed in the olfactory sensory neurons, here in green and yellow, that are randomly expressed in the olfactory epithelium. So every time that the mouse is experiencing an odor, some olfactory sensory neurons will be activated. And the particularity of this olfactory system is that every single olfactory sensory neuron is expressing one and only one subtype of olfactory sensory receptors. So for example, the olfactory sensory neuron in green will express only the olfactory sensory receptor, green olfactory sensory receptor. And all the olfactory sensory neurons presenting the same olfactory receptor will send their projections to one and only one glomerulus in the olfactory bulb. And the olfactory bulb position is preserved across mice. This means that we know exactly for each single odor which glomerulus will be activated in the olfactory bulb. For example, when the mice are experiencing the acetophenone odor, the M72 glomerulus will be activated, while we know for sure that when the mouse is experiencing the carbon odor, a different population of glomeruli will be activated, but not the M72 glomerulus. So taking this information, we decided to use this genetically modified mouse in which the channelrhodopsin, that is a specific opsin that responds to blue light, is expressed only under the control of the M72 promoter. So as you can see from this beautiful representation of the olfactory glomeruli, in these mice, only the glomerulus presenting the M72 receptor, together with the projections coming from the olfactory epithelium, will present the channelrhodopsin and can be activated optogenetically. So if we implant optic fiber right above this glomerulus, we can actually activate the glomerulus in a real-time specific manner. So in fact, that's what we decided to do, was to present the association of the M72 stimulation, only the M72 stimulation, with the presentation of a series of food shock. So every time that we give the M72 stimulation, we give a short food shock, and then with the intent to associate these two types of stimuli in the brain of the mice. So the idea was to completely substitute the presence of the odor with the stimulation of the M72 glomerulus, together with the presentation of a series of food shocks. So ideally, the day after, we want to test if the mouse, in an odor preference task, we want to test if the mouse, how much time the mouse spent in different compartments. So in this task is designed in a way that the mice can freely move from the two different compartments, and in the two different compartments, the mice can experience the two different odors, the acetophenone odor and the carbon odor. So even if they never experienced this odor before, we know that the acetophenone odor is linked with the activation of the M72 glomerulus, while the carbon is not linked with the activation of the M72 glomerulus. So if it is true that we know the code that the brain uses to process the odor information, and if we can activate this code into the brain, we should see that the mice spend less time in the acetophenone odor, even if the mouse never experienced this odor before. And that's exactly what we found. So we found, as you can see in this example of the mouse, speed up, so we can actually really appreciate the time that he spent in the carbon or the acetophenone odor. We can clearly see that the mouse spent less time in the acetophenone compartment during all the period of the experiment that was 10 minutes long. So the mice actually, even if they never experienced the acetophenone before, they were put in this compartment and they actively avoid the acetophenone odor, spending less time in that compartment. So we basically were able to create an artificial memory of this event, the acetophenone, that was never experienced before, a negative artificial memory of this event. And then we ran all different controls to show that the stimulation of the M72 by himself is not actually creating any preference for any of the two odors, as you can see in the second panel. in white here. So the mice spend basically the same time in 2 compartments. And most interestingly, one very important control here is to analyze that when we present the stimuli, the 2 stimuli, the unconditioned shock, the unconditioned stimulus, sorry, the shock 24 hours before the M72 stimulation, the conditioned shock, the mice are not associating the 2 stimuli, and so the mice are not afraid of the acetophenone during the test, and they spend exactly the same time in the 2 compartments. So only when the mice learn to associate the M72 stimulation plus the shock, they do actually, the day after, avoid the acetophenone odor. Most interestingly, here I show you exactly the same results, but here you can see the behavior of each single mouse. And you can see that the majority of the mice that we trained to associate this artificial memory actually the day after have an avoidance of the acetophenone odor. But most interestingly, we found that the same behavior, that there is exactly the same behavior when we create the real memory. So a real memory is when the mouse learn to associate an odor with a series of food shocks. And here you can see that the majority of the mice, in the same way, spend less time in the acetophenone odor. Very similarly to what we were able to create by implanting this artificial memory. And here in white, you can see all the different controls that we did also for the real memory. Naive mice that have never experienced the odor or the shock before. Mice only trained with a series of food shocks. Mice only trained to the presence of the odor, and then mice in which the odor and the food shocks occurred in 2 different days. So they were the mice were never able to associate the 2 stimuli. So the final goal of my experiment was actually to be able to implant a completely artificial memory. So in this case, we don't want to get rid only of the odor experience, of the odor external experience, but we also wanted to get rid of the aversive experience of the series of food shocks. In fact, the experiment that I showed you before showed that we were actually able to associate a real shock with the M72 stimulation. In this case, we also want to substitute the series of food shock with the stimulation of a specific subpopulation of neurons in the brain of the mice. So to do this, we took advantage of this paper from Malenka's laboratory, from Lammels and colleagues, in which they found an input-specific control of reward and aversion in the ventral tegmental area. So the ventral tegmental area is actually an area that we are targeting here. And due to differences in synaptic connectivity in synaptic circuits, they found that the lateral abendula projections to the VTA, if those projections are optogenetically activated, they elicit aversion in mice. While if they optogenetically activate the lateral dorsal tegmentum to the VTA projections, they can elicit reward behaviors in mice. So we took advantage of these experiments, and we tried the same in our mice, in our M72 genetically modified mice. So the idea was to actually induce the stimulation of the M72 glomeruli to substitute the odor experience, and at the same time to stimulate the projections going from the lateral abendula to the VTA. And here you can see in this example in pink, the beautiful projections that the lateral abendula send to the VTA, like a cascade of axons that are reaching from the lateral abendula to the VTA. And we were able to target specifically those projections by putting an optic fiber right above these projections. So we can target specifically those projections, and at the same time, we stimulate the M72 glomerulus. So ideally in this protocol, what we wanted to see is if it's actually possible to completely get rid of the odor experience by AM72 stimulating and by the shock experience by VTA stimulating, and then test the mice for their odor preference test again in this compartment in which the two odors are presented right here for the first time. So they never experienced the acetophenone or the carbon before. So ideally, even in this case, we want to see if the mice are avoiding the acetophenone odor. And what we found is exactly this. So in control groups, we see that when the two stimulation, the AM72 and the LHB2 VTA projections are stimulated, delayed by 1 hour, we don't see any effect. So we see that the mice spend exactly the same time in the two compartments. But when we actually associate with these two stimulations, with these two optogenetic stimulations, we see that the mice spend less time in the acetophenone odor. Again, even in this case, we were able to create an aversive memory of an event that was never experienced before. And in this case, we were able to create an aversive memory where the mouse is just a naive mouse. So it just experienced some optogenetic stimulation, and the day after is without any real reason, basically avoiding the acetophenone odor. It should be a completely naive odor for the mouse, but he's now avoiding it. So then we wanted to see if this is specific for aversive memories, or we can actually create rewarding memories. So in this case, what we decided to do is to stimulate projections from the lateral dorsal tegmentum, again from the paper from Malenka's lab. They were able to stimulate those projections and found that the mice, that these elites reward behavior in mice. And at the same time, we stimulate the AM72 projections. The AM72 glomerulus. So here in the representative figure here, you can see that we inject the virus containing channelrhodopsin in the lateral dorsal tegmentum, and then we stimulate the specific projections coming from the lateral dorsal tegmentum to the VTA. And at the same time, we stimulate the AM72 glomerulus. And what we found that was very interesting for us is that in this case, differently from the experiments that I showed you before, the mice were preferring the acetophenone odor. So we can basically create artificial memories that can be rewarding or aversive due to the type of stimulus, the type of circuits that we are stimulating using optogenetics. So this was very exciting, and in 2016 I was actually still working, like I was collecting the first results for these experiments, and I got really excited during the Canadian Association for Neuroscience meeting. I was at this meeting while I was doing this during this period, and John O'Keefe, the Nobel Prize John O'Keefe, said something about future perspective in neuroscience. And what he said was that if we really think that there is a code in the brain, then we can use it to implant false perceptions, emotional memories. And I think that's exactly what we were able to show in our experiences, in our experiments. And that's why I got really excited about what he thought about futures in neuroscience. And even more exciting was to discover the reaction of the neuroscience community when we shared our results, when we published our paper. And because we believe that we were able to incept an artificial memory in the brain mice. So now things are way much more complicated than the story that I told you before. And of course, to recall, to create, to form memories in the brain, you need the orchestrate coordination of the entire brain that is activated at the same time, that is communicated to each other to actually be able to build and then recall memory traces. So what we wanted to address in our experiments was to try to understand if actually the artificial memory that we created elicit the activation, engage the same circuits that were activated, that are active when a mouse is recalling a real memory trace. So to do this, we trained mice in a real memory and in an artificial memory. And we as well have control groups of mice in which the mice were experiencing only the odor, in the case of the real memory, or they were only stimulated by M7. They were only receiving the stimulation of the M72 glomerulus. And then we analyzed the different pattern of neuronal regions that were activated to recall the memory trace. And we picked the different brain regions in the brain due to their contribution to olfactory processes or their contribution to aversive processes. And what we found is that in both real and artificial memory, the type of circuits that are activated were very similar. And in particular, we focused on the basolateral amygdala. The amygdala is a very well-known region to be involved in the association between a conditioned stimulus and an unconditioned stimulus. In our case, the odor and the food shock or the reward. So we know already that the amygdala is very important for this association, for make this association possible. And it was very exciting for us to show that even if we activated only two tiny points into the brain of our mice, we were able to induce the similar pattern of activity in different brain regions, and in particular in the amygdala. So in the amygdala, we found that there is an increase related to the control group of the amygdala activation in both real, as we expected, but also in the artificial memory. So we wanted to test if this amygdala, if the amygdala activation is important for the recall also of the artificial memory traces. So we first tested again the contribution of the amygdala in real memories, and we confirmed the previous works that shown that when you create a real memory, like for example the association between an odor and shock, and then you inactivate the amygdala right before the recall of this memory trace, mice actually forget the memory association. And in this case, they spent the same amount of time in the two compartments. So these are just this is just confirmation of previous works. But what we were very interested in demonstrating is that even in artificial memories, when you inactivate the amygdala right before the recall of the memory trace, you have the same results. So here what we did was to train again the mice to create an artificial memory by stimulating the M72 glomerulus with the LHB to VTA projections. And 1 hour before the recall of the memory trace, we injected a drug using a chemogenetic approach. So in this case, we inject a specific virus in the amygdala, and then we can inactivate 1 hour before and for the entire duration of the test, the amygdala. And what we found is that also in artificial memories, when we inactivate the amygdala right before the recall of the memory trace, mice completely forget about the artificial memory, and they spend exactly the same time in the acetophenone versus the carbon compartment. So it looks like the amygdala, even in this case, is important for the recall of the memory trace. Even if we never touch, we never activate, we never manipulate the amygdala to create this artificial memory. So in our laboratory today, we are interested in analyzing these codes that the brains use to create specific behaviors, to incorporate in our brain specific information, external information. But we are also interested in how those informations that we acquired from the world outside us are then consolidated into the network, and how the specific codes in the brain are reorganized over time. Because memory is like an active process that allow that continuously modify the original memory trace and create a completely different memory trace from the original one. So we are now interested in understanding how these specific codes that the brain uses to incorporate external information are then changed over time to be recalled later on, years after the original trace was created. And we are interested in analyzing it at different levels, from a nuclear level to a synaptic level. So what happened at the synapses that are the most important connection between neurons, how the neurons communicate to each other, to a cellular level. So what happened at the level of a cellule, and how this can be incorporated in the population of cells, how the population of cells changes their activity over time to consolidate a previously acquired previously acquired memory. And then finally we move to the network level, so to see how different brain regions are coordinating their activity to recall the memory trace. So in particular, at the now in our laboratory, but eventually combining these techniques, these very interesting techniques like optogenetics and miniaturized microscope, we intend to be able in the future to decode specific memories in mice. And with this, I want to thank all the people that were involved in the experiments that I showed you today, and in particular, the C4 team that is growing every day more. And thank you for your attention. And of course, I will take all the questions that you have.
So can you hear me?
Yes, I can.
Is it working?
It is working. Yes, I can hear
great.
you.
So thank you very much for this great talk.
Thank you.
I'll be transmitting the questions from a member in the chat. So I start with the question from Mehdi.
Okay.
He's asking, from an evolutionary point of view, when did the concept of memory appeared, and how is the memory of mammals and humans in particular specific in comparison to other beings? How do you I guess you asked how do you translate memory from one animal model to another?
Yeah, that's very interesting questions. I mean, we are studying very basic memories, and that's a very important point of my talk. I'm not saying that we can actually create artificial memories in humans. That's impossible, and that's also ethically incorrect. And but what we are actually studying here are specific, very simple memories and the circuits that are involved in these memories are the same that are involved in the memories in humans. So we are just taking very, very simple and very straightforward types of memories, type of stimuli that we can bring back to the study of humans. Can you hear me? Can you hear me?
Okay, thank you very much. Yes.
Okay.
Can you hear me again?
Yes, yes, yes. There is a little bit of a delay.
Yes, Gisela, I had a small question.
Mhm.
So, do you think we could reverse innate fear into something more neutral or positive, even though it might be like hardwired? For example, if you take mice and rats are afraid of the odor of urine coyote
Yeah,
urine, do you think we
yeah,
could
yeah.
reverse it?
That's a very intriguing question. I mean, that's something that intrigues me the most, to try to understand what they are thinking when they actually recall these artificial memories, because I'm sure they don't have a conscious experience of this odor. They cannot really tell, okay, this odor reminds me of yesterday that I get the shock. So I'm not sure what we are creating are innate memories. You know what I mean? It's like something unconscious that brings you to go further from that odor that you never experienced before. You don't know why, but you want to avoid this odor. Is this like, are we actually creating an innate memory? So to answer your question, yes, I think it will be very interesting to try to revert, to change an innate memory, a very strong memory, in this kind of approach.
Yeah, but my question is a bit more complex because I don't know if, for example, we are talking about innate fear when we talk about the coyote urine, but is
Yeah.
it really a memory or is just something that is hardwired like a reflex, for
Yeah, yeah,
example?
yeah. So those are memories. It's very complex. There is like all literature talking about what is an innate memory and how we can distinguish it from like a learned one. I mean, it is a very complex. It's very difficult to. We don't know which circuits basically are more involved in the two type of memories. There is a lot to learn about it, but that's absolutely something that I'm very interested in.
Okay. Thank you. Jeremy, do we have other questions?
Okay, so we got another question. Do you think the remaining questions are mostly fundamental scientific? Yes. Do you have other questions? Are you hearing me?
Yes.
Okay, great. Sorry, I got the delay.
Okay.
I'm going to re-ask the question. I have a bit of delay. So according to your current results,
Yeah.
do you consider the head direction cells as a substrate for memory, trace, or as sensorial cells, or cells that got another role?
So they are for sure sensorial cells. They are that's the reason why I'm very intrigued about the head direction cells, because they are one of the most sensorial cells. I mean, that we can find into the brain. When the mouse is in that direction, they are gonna fight. So it's very reliable, their activation. But it has been shown that they are also linked with memory. So if you train the mice over and over again in that context, these cells learn, what is their head direction. So it's a bit of combination of the two, and that's why I'm very interested in analyzing them over time, because at that we will be able to see if their behavior changes due to a memory that's linked with the specific context that they learned days before. So it's very it becomes now a memory, a special memory, and now we can study how those cells changes their activity over time. Can you hear me?
Yes, we can hear you.
Yes, it's working. So does anyone ask other questions? You can ask them in the chat, don't hesitate.
And Gisela, I was wondering, why do you think the mice, they don't experience the memory like it would be really real? I don't know if my question is clear, but if you activate the exact same circuit that are supposed to be activated during the circuit, why do you think the experience of the mice is different from reality?
Oh yeah. That's a good point. I mean, what I think is that I'm not giving them exactly the same experience because we don't have the exact knowledge of how our brain works. Example for the odor, we actually give just part of the experience of the odor. The acetophenone is constituted by the activation of a complete, like a very complex number of glomeruli. We are activating only one of it. Even though we can create this artificial memory, even though the mice is like still avoiding the acetophenone odor, we are not recreating exactly that experience. And I would say that to recreate exactly that experience, you should not activate only one cell or one subpopulation of cells in the brain. You should be able to recreate what happened into the entire brain when you have this sensory experience, and that's way much more complex than what we have. That's why my feeling is that the mouse is not really feeling exactly the same.
And the mice is not sniffing at the time she's
exactly. That's another
smelling the
interesting thing.
odor
Yeah.
also. Like she
She's
smells,
not,
but she's
exactly.
not trying to smell. So
Exactly. And even in the case of the aversive experience that we simulate with Optogenetics, we never see the mice like really having the fear, experiencing the fear, the pain. We can, in this case, activating this kind of circuit, we cannot have that kind of behavior during the training. We still see the creation of the memory, but we are not recreating exactly, everything that happened into the brain when you have a shock experience.
okay.
We are activating only part of it. So
When you're stimulating into the Vetere?
exactly. There are way much more like
it's like having the bad side of suffering, but without suffering.
Yeah,
because they don't feel pain. Okay.
Exactly. And that's why we want to try to use this kind of techniques to revert eventually negative memories to positive memories,
Because in the Vetere you could also stimulate for pleasure, right?
Exactly. And that's what we are aiming in a different set of experiments for the future. We would like to create, to revert real memories, real negative memories, and target only the specific circuits that are involved in that specific memory without touching the rest of the brain. That would be very interesting.
Okay. And I had one last question, Gisela.
Yeah.
So when you're stimulating while the mice are awake, right?
Yeah, exactly.
What would you expect if you stimulate them only when they sleep? Because there is a bunch of literature talking about consolidation and reconsolidation of already made memories. But do you think incept the memories only during sleep?
Yeah, that would be cool. I mean, we tried that. I would say it was not during sleep; it was during anesthesia. So we never succeed to that. But I mean, I didn't try. It was not working at the beginning. I don't know. Maybe we should have changed the protocol to make it work. But I think it's possible. It's totally possible. This may interfere, like if you do it during sleep, this may interfere with the consolidation of other things that they are like consolidating at that moment. But that may be very interesting, actually.
Or maybe it's not possible because you don't have any I don't know grand truths to reconsolidate during the sleep if you haven't started the manipulation first.
Yeah, that's a good point. Yeah. And it may be that he also didn't work during our anesthesia because of that.
Yeah.
But that's something that we thought about because we wanted to be the mouse completely unconscious, and but that's something that we never managed to do.
Okay.
Okay, so if everybody's done with the questions, I'd like once again to thank you very much, Professor Vetere. I believe everybody enjoyed very much this talk.
Thank you.
I hope you enjoy being with us today in those peculiar circumstances.
Thank you.
And we'll be welcoming you to all the attendants. We'll be welcoming you next Saturday with John DeVos that will talk. That will give his talk at 4 p.m. Much similarly than today on the same platform. And well, we'll be waiting for us. We will share information regarding his talk this week on a new event. It will be about his work on pluripotent stem cells. So once again, thank you very much. Thank you very much, Professor Vetere.
Thank you for inviting me.